The first one we'll talk about is an old-fashioned one, but one that's actually perhaps the most easily quantitated. Okay? And this is called filter binding. Okay? Basically, you use a positively charged filter paper. Okay? Typically, it's something called DE81 paper. You can also use nitrocellulose and several other types of filter paper as well. Okay? You spot the reaction on the paper. And one of the nice things about this is that that actually kills the reaction very quickly because that will rapidly dry, and a dry reaction can't work. You need water for the reaction to go. Okay? And then you can take this material and you wash with buffer that has salt at a level that releases DNTPs but not DNA. Okay, so why does it distinguish between the two? The answer is actually pretty simple. How many negative charges on a nucleotide on average? One more, 3. It's 1 per phosphate, basically. Okay? So typically, although there can be, for short periods of time, more than 1, on average, you're going to have 1 negative charge per phosphate. Okay? So a DNTP has 3 negative charges. How about on our DNA substrate up here? You don't have to be exact. More than 3? Lots, right? I mean, even without any synthesis, that has more than 5,000 negative charges because every base has a negative charge associated with it. Okay? So DNA is going to be, let's just say, thousands. Okay, and let's make the argument that in order for a molecule to be released from the filter, it has to have all of its negative charges released from the filter simultaneously. Okay? So the salt will do that because it will come in, and when it binds, it will take the place of the positive charge on the filter. So let's say we use sodium chloride, the sodium ion will come in there and take the place, but it can only do so many at a time. Okay? And depending on the concentration of salt, that's going to happen more or less fast. Okay? But when you only have 3, right? Let's say every, 2 or 3 milliseconds, one of those charges releases, and that's happening sort of on average the same for all charges. Okay? Well, that means you will have during every, say, 5 seconds or something, all 3 of these charges will release at some point simultaneously. Okay? But the odds that 5,000 of them are going to come off simultaneously is essentially 0. And so you just wash these things. Usually you do it 3 times. You can imagine they're just little discs like this. You throw them in there, you wash them 3 times. Usually you do this with radioactivity, and so you can take the supernatant. You can say, do I detect any radioactivity in there? And you'll do it until you don't detect any radioactivity coming off the filters, and you're done. And then you merely take that material and measure the amount of label associated with it. Okay? If it's fluorescent, you can't quite do that as easily, but the same idea holds. 3 washes, you're done. Okay? It's very easy to get this to work. Okay, so this is easy to release. And this is hard to release. And if you choose the salt right, hard becomes impossible. And typically you do this in like 50 millimolar NaCl, and this will come off right away. This guy will never, ever, ever come off under those conditions. Okay, you probably need 2 or 3 molar salt to have a hope of getting something like that off. Okay, so what do you do with it at this point? So you basically take this material and you count or measure the fluorescence or radioactivity associated. Okay? And that gives you really down to the femtomole or lower attomole amounts of nucleotide that's been incorporated. And if you have a good understanding of what the amount of, for example, radioactivity or fluorescence that's associated with your starting nucleotides, you can measure this to very high precision how many nucleotides have been incorporated into your template. Okay, so the advantage of this approach is it's fast. This whole thing probably could be done in about 8 minutes. Okay, if you don't count the scintillation counting or the way you measure the fluorescence, it's incredibly fast. Okay, the washes take 30 seconds each, and you dry it and you scintillation count it and you're done. Okay, and it's also quantitative. Okay, so this is probably the most quantitative you can be about, for example, how much nucleotides did you add over how much time to how many templates. Okay, so it's nice in that way. The disadvantage, which you may have figured out by now, is there's no information about length. You would see exactly the same result in this assay if you made 1,000 10 base pair extensions or if you made 10,000 base pair extensions. No way to tell the difference by this assay. Okay? Everybody understand that? You're just counting how many nucleotides are incorporated. You can't tell what the nature of the product is. Okay? So this assay, I'll tell you, is typically used when you're just fractionating and you just want to know where is there a polymerase, because it's really fast. If you have 100 column fractions from separating out, for example, a crude extract of some cell, you can use this to very rapidly say, okay, here are the fractions that have any DNA polymerase activity. Okay, that's basically where this is used.